During this practice, the ligation of a PCR product to a topoisomerase coupled vector was carried out, for which it was necessary to know the main constitutive elements of the PCR 2.1-topo vector used, as well as the molecular characteristics of the PCR product to ensure the complementarity between the elements of the ligation reaction and consequently the transformation of the bacteria.
Cloning with PCR 2.1 topo is based on the capacity of ligation by the topoisomerase. When the plasmid which poses the enzymes at ends is in contact with a PCR product having adenines, the topoisomerase performs ligation of the phosphodiester backbone. It is subsequently destabilized and cleaved from the plasmid. Then for the ligation to be carried out, it's necessary the presence of thymine and adenine for the ligations. This vector is useful for rapid cloning because it does not require other enzymes such as restriction enzymes or ligases, and its efficiency are high. It is also useful for in vitro transcripts using T7 promoter, and due to its linearized form is useful for qPCR procedures. These are the features of the PCR 2.1 topo plasmid. The PLAC is a promoter for LAC-alpha. LAC-alpha encodes for beta-galactosidase. The F1RE is an origin for single-stranded replication. Kanamycin is a kanamycin resistance gene. Ampicillin is an ampicillin resistance gene. The PUC-RE is a high copy origin. There are many topo vectors. In the table are shown 3 of them. PCR4 topo allows for more insert sequence and less vector when using standard sequencing primers, and a protocol amplification of 5 minutes long with 95% of efficiency. The PCR plant 2 topo allows direct selection of recombinants, easy excision of the inserts by restriction sites, and it has primers for sequencing or PCR. PCR XL2 topo has a positive selection with CCDP, allows for cloning PCR products up to 13 kilobases. To carry out a ligation, it's necessary to take into account the protocol that will be carried out and the concentration of salts. The concentration of salts for chemically competent cells should be adequate to allow the neutralization of the charge. On the other hand, in a protocol for electrocompetent cells should be avoided very high salts concentration because the voltage supply can generate electrocution of cells. Finally, it's important to know which cells are correctly linked. In order to differentiate the colonies that have the vector with the insert, a selection is made by XGAL. This selection is possible due to the alteration of the ORF of the gene that codes for the beta-galactosidase. As the information of this gene is interrupted by the insert in the plasmid, the bacterium is no longer able to synthesize the beta-galactosidase, which degrades the compound XGAL producing metabolites with blue coloration. Therefore, the coloration of the colonies that present the insert inside the plasmid will be white. To verify that the colonies have the insert, it can be performed protocols as mentioned, blue-white coloration, PCR, or even using restriction enzymes.
From the results, Figure 1 deposited control LV medium plus XGAL. From the results, Team 3, the Figure 2, a polygraph in LV medium with PCR topo plus XGAL plus ampicillin incubated for 24 hours at 37 degrees Celsius of electrocompetent cells. They were observed 0 blue colonies and uncountable white colonies. From the result, Team 4, Figure 3, a polygraph in LV medium with PCR topo plus XGAL plus ampicillin incubated for 24 hours at 37 of thermocompetent cells. They were observed 0 blue colonies and white uncountable colonies.
A PCR product ligation using the PCR 2.1 topo is fast and easy to perform. The key to topo cloning is the enzyme DNA topoisomerase 1, which according to Shannon functions both as a restriction enzyme and as a ligase. Its biological role is to cleave and rejoin DNA during replication, therefore in this case reduces the reagents needed. These vectors are provided linearized with the topoisomerase 1 covalently bound to the 3' phosphate on each end, which enables the vectors to readily ligate DNA sequences with its compatible ends. Therefore, PCR products that present an adenine residue on its end presumably affect its interaction, leading to its separation.
The plasmid presents certain elements that have an important function for the correct development of the product ligation and subsequent cloning into a given strain. These elements are shown on this figure. The PUC origin is the bacterial origin of replication, allowing multiple copying once being on the selected strain. That is shown in here. In this case, the lag seed fragment codifies for the beta-galactosidase fragment and also acts as a multiple cloning site. Both M13 forward and reverse primers are primary union sites useful for sequencing or PCR screening that are here and here. The T7 promoter also acts as a priming site for in vitro transcription, that is in this case in this part. And the F1 origin is spheric and allows the synthesis of just one strand during the replication, therefore being faster. It also includes two antibiotic resistances, kanamycin and ampicillin, for clonal selection, and all these features allow a high-efficiency system for ligation products and cloning. When choosing the correct strain, it is important to consider that it does not degrade heterologous DNA. In this case, the plasmid vector of topo. Therefore, L. coli DH5-alpha is commonly used as it genetically is modified to suppress its reaction to degrade foreign DNA machinery, disease their endonucleases. It also avoids the recombination through Reg-A mutation for an easier extraction. With respect to the kind of ligation that is made in this part, there are several problematics that can be shown, mostly because of the specificity of the ends from the vector available for ligation. This is because they are not specific for each end from the PCR product. Therefore, they can be joined in the missense fragment and with an incorrect ligation. The first of these variations is the high-fidelity blunt-end topo cloning that implements the high-fidelity polymerases that avoid the adenine ends from contact polymerases. With this method, blunt ends are generated due to their exonuclease activity, and then ligation is performed with blunt ends in the vector and the PCR product. The second variation is the directional topo cloning that enables cloning of blunt-ended PCR products directly into an expression vector in a single orientation. In this case, the vector has a sole GT-GG region overhang which invades the double-stranded DNA for the PCR product and anneals to the GACC sequence placed on the primer for the PCR product, being then specific for just one side of the vector and directing the PCR product site of ligation to it.
There are several ways in which we can verify that the correct ligation was made. One of them is the verification by amplification using the M13 primer sites on each side of the multiple cloning sites. Also using a fragment of the lag gene that is also in this part that enables discarding for the incorrect ligation products visually, and also sequencing on the construct that may ensure the correct orientation of the fragment, and this can be performed with the M13 primers. Also, when there is a 100% of transformation yield, there may also need a verification, so these methodologies can be used. Therefore, as the total transformation yield can be possible, it could also represent incorrect reagents activity such as the non-functional X-gal or the missense construct presence. For a correct ligation, they may be verified with sequencing or enzyme restriction. Finally, it's important to mention that during this development, a previous transformation was made in order to determine how competent were the cells prepared for this methodology. In this case, there was a supercoid DNA used, which had a high efficiency for both cases. But in here there was a slight difference, as, as it has to be considered that the vector was linearized as it comes from the kit. So this slight difference is important to take into account in order to determine the efficiency of the experiments made.
First, we have to thaw the PCR product on ice, as well as the reagents of the kit in ice. Then we have to prepare the ligation reaction for chemical transformation or electroporation, depending on the cell we have chosen from the last practice. We have to save the ligation at minus 20 degrees. We have to transform using 3 microliters of ligation into 50 microliters of chemicompetent or electrocompetent cells. And then we have to incubate them at room temperature for 10 minutes. We have to remember that to prepare the ligation, we can use the quantities of the reagents shown in the table below. After transforming, we have to add 300 microliters of sugar. We have to spatulate the entire volume on an LV plate with ampicillin, and after that we have to incubate again with the plate at 37 degrees overnight. The next day, we have to take a picture of the plate and store the plate in the refrigerator. With this, we have to receive 2 colonies, one white and one blue, of those that grew on the LV plate with X-gall and ampicillin to facilitate extraction of plasmids and verify the presence of the insert by digestion with restriction enzymes.
In conclusion, there was no growth of isolated colonies to calculate the transformation efficiency, nor of blue colonies to make a selection of recombinant bacteria. That is, the growth in both the negative control and in the plate with the ligation product was of a white coloration. This indicates a growth of cells with a plasmid in a medium in which the antibiotic is degraded or that the X-gall reagent was not in optimal conditions. For the topoplasmid ligation to be successful depends on multiple factors. However, there are specific points that have been characterized as critical to ensure the success of the reaction. The first is to establish what quantities of plasmid and PCR product will be added. For this, it is important to take into account that a 1-to-3 molar ratio is required, as well as the specifications of the kit provider. The second, the selection of the type of topo kit that will be used depending on the ends that the insert acquires after the PCR to ensure the complementarity of the ends of the vector with the insert. And third, it is important that the cells used for the transformation are suitable, that is, that they include mutations in genes of nucleases and recombinases in such a way that they do not interfere with the transformation, the replication, or even in the transcription of the plasmid. Likewise, it is important to perform the analysis of the ligation product by techniques such as colony PCR, restriction enzymes, or sequencing to determine the presence of the insert and its correct orientation.